Hi everyone, I'm Jeanette May, a field application scientist at Countable Labs, and today I'd like to walk you through how to set up a Countable PCR assay using just one tube from beginning to end. If you'd worked with qPCR, or especially with digital PCR, you know that it can take weeks to get your assays up and running reliably and reproducibly. There's often a lot of manual pipetting involved, and you lose a lot of material to dead volume when you're doing sample handling for dPCR. Plus, it often takes days to get the replicates and controls that you need to quantify your genes of interest. With Countable PCR, everything happens all in the same tube. This makes the protocol super easy, free of pipetting errors, and free of microfluidics that can introduce this sort of dead volume, which also makes it possible to do single molecule counting. There's no multi-day workflow, unlike other technologies, or long turnaround times to analyze the data. The technology we have is super powerful, yet simple enough to work in a single PCR tube. So how it works is that we start with a mini spin column that goes inside of a PCR tube. You load your PCR mix into the spin column, and then you put it in a standard Eppendorf benchtop centrifuge. After 20 minutes of centrifugation, the target molecules that you're looking for gets dispersed into the tube and encapsulated in a unique matrix. Here's a blown-up example of that matrix. Each of the wavy lines here represents a DNA target isolated by itself in an independent compartment. The matrix contains the PCR mix, primers, and probes, all dispersed in this matrix along with all the targets. During amplification, in a standard thermocycler, the signal from the single molecule gets amplified in the color that you specify with your probe chemistry. Because the amplification happens by itself in a single molecule compartment, multiplexing, or amplification of multiple targets in our platform, is quite simple. There's no concerns about amplification bias. And then detection and counting are also equally as easy. You simply take the same PCR tube and place it into the countable instrument for imaging. The instrument scans to the tube directly with hundreds of light sheets and then provides you with the true absolute count for each target in each channel. There's no standard curve needed and no statistical Poisson error correction, unlike legacy platforms. It's simply the number of targets in the tube itself. I hope that shows you how easy it is to set up an experiment with single molecule counting of your gene targets. With this straightforward PCR workflow, you get high precision sensitivity so that you can get the most confidence in your results. If you want to learn more, you can connect today with the expert at Countable Labs to see how you can get started with Countable PCR.